Hello!
Hi,
Welcome to YouTube Microbiology Journal Club, where you nerd out big about all things small.
my name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. But nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to enabling the public with the tools of
And my
biotech.
name is Faz, and in a previous life, I got my PhD in microbiology from Imperial College London, where, amongst other things, I was making flesh eating bacteria glow in the dark. But now I've moved on. I've worked in other jobs, such as a research integrity specialist, but now I'm a manager for an academic journal.
And every week we meet to talk about microbiology. This week it's our deep dive week where we look at each figure in a paper that we chose from last week and learn to criticize the scientific findings together. This is a journal club, and we encourage everyone in our audience to leave us questions and comments.
Next week will be our news week, where we survey a bunch of articles to choose an article for our next deep dive. So make sure you subscribe to satisfy your microbiology curiosities.
And you can follow along with the papers that we discuss on either week in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet at us using the hashtag microtwc hashtag, which is down there. Yep. Uh,
somewhere in this overlay.
Indeed, yes. Uh, so yeah, this week.
So this week we got a paper titled mRNA-1273 efficacy in a severe COVID-19 model, attenuated activation of pulmonary immune cells after challenge.
Yes, so vaccines are being rolled out everywhere now, and evidence has been gathering that they can reduce severe COVID infection, and some signs that they can also uh, having effect in covering the outbreak. Um,
Yeah,
yeah,
totally. And I think when we chose this, we were very excited because like vaccines are like done with clinical data, right?
mm-hmm.
Like is how we often get vaccine stuff. It's like you see the efficacy of it in reducing disease, but those finer mechanistic points, like how does it really work in the body, right? What cells are doing what, like what molecules are being activated, that information is more difficult to come by, and this seemed like a really good source of that sort of information.
Yeah, I mean, now that we kind of know that the vaccines work, this is delving into like how they work, and because with the vaccines we aim to train the immune system to respond to SARS-CoV-2 the right way, and we've covered papers where we've seen how SARS-CoV-2 like infection can make the immune response the wrong way. So
Hmm.
so here this is going into like so usually we get like antibody data, we get some cell data, but we don't really get an idea of what the entire immune system is doing. Um, so, yeah, and it's quite hard to get that sort of data because in some cases you have to like pick out every single cell, and that makes ethics committees very like unhappy if you do that to a whole person. So, um,
Yes, I mean, I guess like biopsies are difficult to come by, right?
yeah,
On
we
people.
we, we
Yeah,
covered
we definitely don't see that sort of
we've,
stuff.
we've covered exactly only one paper that's managed to do that, and that's had to use cadavers from people who had died of COVID, which means that we've
Hmm.
gotten a much more greater understanding of like what happens in severe COVID. But what about the other COVID outbreaks? What happens? What does a good immune response look like compared to a severely bad one? And this is something
Hmm.
this paper will aim to answer. Um,
Hmm.
so let's move on to the slides. So yeah, this paper has everything. It has lungs, it has mRNA, it has a bit of machine learning to discover what cells do. Um, so let's move on to like a bit of background.
Yeah. So, I mean, I guess they're going to be using an animal model here, and so I think I wanted to remind people that usually animal models they're limited because the immune system has to match something that we're seeing in humans, but also they have to be able to get the infection in the first place. And a big roadblock into getting the infection is the receptor to the virus, which is ACE2. And so here we have a comparison between human ACE2 and a whole bunch of other creatures. So you can see actually it's very similar. The receptor binding domain is really similar to the macaque, rhesus macaque. We've talked about that model in the past. It's pretty similar to hamster, which is a model that we've also talked about in the past. I think we've talked about a lot of these
Yeah.
models. It's pretty similar to ferret. I think ferret is actually something that has been a model. We have never talked. Oh, we saw it briefly in the transmission study. They did ferrets.
Yes.
Um, and mouse is actually quite different. Now we're kind of moving to even more dissimilar. And we've definitely talked about mouse before. They don't get COVID
Mm-hmm.
when you just give them the virus unmodified, but you can do things like modify the mouse or modify the virus to make the disease happen.
Yeah. So moving on to the next slide. So we've seen people use rhesus macaques and primates, which are very close to humans, and they they do have a very like kind of. So here we've got like an X-ray that shows kind of lungs get some infiltrates in them. We've got
Yep. So that's like pneumonia, right? They're getting pneumonia of some sort.
yep, which is somewhat similar to what we see in humans.
Yeah. They get like a decrease in body weight. They get a little spike in fever, body temperature. And then this is just a big table like that has all the different things that you can see.
Sorry,
It's
I
pretty
just
tiny.
need to double check because ages ago we had a lost episode where we were covering like a pneumonia model in macaques, which was looking
Yeah.
remdesivir, and the images for the uninfected and infected were both the same, which is why we never really
Yes.
published that video, I think. And and I was just checking that we
Same,
didn't.
same, yeah, same group, different images.
Yeah, I was checking to see whether they'd made that same error because that'd be embarrassing to be caught out on stream like that. But um, Uh, so that's why I was like squinting up
No,
really close to the camera to see whether. Um, but yeah,
I thought maybe you're trying to read some of the clinical signs. Anyways, in the clinical stuff, you can see they get it, and actually a bunch of them, like by day 21, observations at like necroscopy, where they end up sacrificing the animal. By that time, like they're fine usually, like they recover, right? I think you can see.
yeah,
It's hard to read, but all four macaques that made it to day 21. One of them recovered on 9 days post infection, one recovered 10 days, one recovered 17 days post infection, and 13 days post infection. So, like, the way that they challenged these animals, they did not get a severe disease.
yeah. I think that's and also I think we should recognize that for a lot of humans that get SARS-CoV-2, there is a certain proportion who don't get a severe disease as well. So there is that.
Absolutely.
You're always going to be rolling that dice when you're doing a model with this kind of disease. But
Yeah.
gen-
And I think that, but like, because I think it's important when we're thinking about vaccines, right? That vaccines, the big work that they're doing is preventing people from getting severe disease,
hmm,
right? Like that's a major clinical outcome that we care a lot
yes.
about, like as we use them. So when we want to study how the vaccine might do that, we have to make sure that our model can get a type of disease, right? That is somehow analogous, or we can make some sort of analogy, right, between the human form and what we see in the model.
And I think that brings us to hamsters. Um,
Yes, which do get this. So they can bump up the amount of virus that they give into hamsters, and you actually get a bunch of the hamsters to die.
yeah, and there's a number of reasons why I'd rather use hamsters than primates because they can both get the infection, but also primates are very hard to keep. They're very intelligent animals, so you need to really put in a lot of work, and they're very expensive. And also there's humate. Whereas hamsters, you can keep them happy for a lot less money. Um,
Hmm.
and that means you can use more of them, so you can get more numbers, and then you can have a higher chance of seeing the variety of diseases. So with any like kind of
Yeah.
study, you're not just looking for values, you're looking at the variances. And so the more animals you get,
Yeah, like the biological replicates, right?
yeah,
Like because biology is really variant, so like you want to be able to repeat it over and over again. You can't really do that many repetitions with a with an animal, with a monkey. I mean, I think that may also be part of the reason why they didn't do like some ridiculously high challenge on those
mm,
macaques, right? Because like that's not the niche of animal research that macaques are filling, right? They're not made to like burn through 20 of them, right? 30 of them or something. That's not how we
yeah,
treat that resource of the animal.
yeah.
um,
I mean, we already don't really want to be doing research on macaques. It's just like if there's no other solution, and even then we want to make sure that we use as less as possible. Um,
right.
and
So,
yeah,
anyways, they have this model of hamsters that they use, and I guess what's interesting is that it was even shown that it responds to vaccination,
yes,
right? So they vaccinated with adenovirus-based vaccine, and it was able to reduce the mortality in their hamsters. So we know that in terms of vaccines and mortality, this model captures a little bit of that. And I just wanted to point out here, there's this little side chart where they also correlated that with the neutralizing antibody titers they're able to make in these animals.
yeah. So basically, the more vaccine, the more antibody they get, basically, and the more,
Hmm.
yeah, the more safe the animals get. Anyway, so they're testing it on the Moderna vaccine, so mRNA-1273.
Yep.
So
Yeah. Moderna vaccine. This is as we know, it's a lipid nanoparticle. It has mRNA in it. Somehow that gets into your body, and we've seen the phase 3 trials, so we know that it works like for the clinical outcome. And we know in phase 1 and 2 studies there are neutralizing antibody titers, but beyond that, we can't connect mechanistically through the science, through data support, I guess,
mm,
support into the data, how exactly this immunity comes to be.
yeah. Um, so I think that so we know that since antibodies are made, there is something there, but so the authors have decided. So this is like kind of the structure of how this study is going to progress. So we're going to be. So this is like kind of a plan. They show all the hamsters and all the treatments they're going to get. So you've got your
Yep.
uninfected group. And you've got your mock vaccinated that will later be infected. Uh,
Hmm.
so and then you've got various different dosing strategies they also test. The
Yeah. Yeah, they do like just single dose. They do one dose with a boost of a different amount. Yeah.
Yeah, and also they list their measurements. So the main outputs they're going to be looking at. So there's weight, which is a pretty simple thing to measure. We can talk about that more. Viral load,
Mm.
that's something that we all care about because how much virus is there. Pathology, so looking at taking slices of lung or whatever on a microscope and look at it and seeing
Mm.
what are the cells actually doing. And last one is scRNA sequencing, which is a really brand new amazing technique that we can go into a little bit now, but we'll talk more when it
Yeah,
comes.
we definitely have some slides later.
Yeah.
But yeah.
Um. So
So one of the reasons we chose this paper originally was because we wanted to know where does the Moderna RNA go, right? Because we know it goes into cells, but there's actually I couldn't find it's very difficult to find information which cells it goes into. Unfortunately, this paper because of this the design of when they do their sampling, they're sampling like after the infection, right? They're not sampling directly after the vaccination moment, and so they don't know where they don't end up looking for that information, like where is this mRNA in the cells. um, they must get into the cells because that is how, right, and that's how you could possibly get the spike to be expressed. So we know the mRNA has to get into some cell, right, and that cell has to make some part of the spike protein, right? Otherwise, how would they have seen that antigen, right? Like that much is certain, but the details of it are kind of unknown.
yeah.
Um,
But I guess it's interesting to think about just like how much the cells change when a virus or a vaccine attaches them because we'll be the thing about this paper that's unique that compared to like how we'd be doing this like even 5 or 10 years ago is that we can actually look at each of these processes that are shown on this graph like through the mRNA and kind of and use that to profile the cells and see what's actually going on in them. Uh,
sure, you're talking about the single cell
yeah, single
technology,
cell
right? Is that
technology,
like, yeah?
which we're gonna have a brief overview of here, which because essentially it separates out each single cell and and it's set up in a way that it sequences all the RNA for that cell, and then puts it into a
Hmm.
machine, and the machine can magically like pick out each individual cell, what RNA that cell has, and figure out what it is and what it's doing.
Yeah, absolutely. Yeah, it's able to separate the cells, and we'll review it again, right, when we get to that piece of data.
Yeah.
Uh, but it's a new technique. It's a almost a natural evolution coming from all this next generation sequencing stuff that's been going on, right? Like, I'm sure our viewers probably know it's like the price to sequence a human genome is going down by leaps and bounds, right? Like there's DNA everywhere. If you can find it, you can tell what might be living there. Even at the beginning of the SARS outbreak, they were saying like, oh, we could use this technique to monitor environmental
Mm.
SARS, and like, right, we could swab everything and tell you like all the different surfaces that might have RNA on it. So there's a lot of infrastructure and work that's being done to process those large amounts of data. And with a slight modification, you're able to narrow down to single cells and say this cell is making this particular sequence. Okay, but we'll cover it more when we actually get to it.
Yeah, that brings us to the first figure of the paper. Where our first stop is always with the antibodies. Antibodies are if you've
Hmm.
seen our show, you've probably seen hundreds of graphs like this, where
Yeah, tons of ELISAs, or yeah, these are ELISAs.
Yeah, so they show us every data point, which shows good. I always like it when graphs show us each data point, because then we can get an idea of like where each
Mhm.
animal is, where each individual in this population is in terms of where their antibodies are, and they separate up into their groups. So we've got our like our like uninfected, sorry, our mock vaccinated group, which have no antibodies.
Mhm.
We've got our
Mhm.
single dose vaccinated group. We've got our like 2 dose vaccinated group. Then we've got like two low dose groups. One where it's like 20, like a fifth of the dose, and then like so one's 5 micrograms, one's microgram, and the baseline dose is 25 micrograms. Um,
Yeah.
so
And there, it's actually surprising how similar I think the 25, 5, and 1 are to each other. um, they do have some statistical differences, but yeah, the groups actually to me look quite similar.
yeah,
Again, 1 microgram seems like it's a little bit weaker, but between the difference between 2 and 25 in the prime boost, um, yeah, actually there is no
so
statistical difference I think between, or maybe they never
I
measure
mean,
it between.
I have kind of have an they do kind of measure the statistical differences. They do like 2 different like kind of post tests, and they do some fancy statistics.
Mhm.
One thing I'd like to kind of point out is that the human dosage of mRNA that we get from Moderna is 30 micrograms, and giving
Mhm. Mhm.
a hamster 25 micrograms is very similar. But then again, it's a much smaller animal. There's a lot more.
Yeah, I was gonna say it's like I don't think it's very similar at all when you think about body size.
Yeah, yeah, exactly.
Yeah.
I mean, one immediate point I have to something that always have to consider with any animal study is the science of allometry, where like the dose you give to a human might not turn out the same for an animal that's smaller. So for
Mhm.
I mean, I think there's this Haldane quote that I was thinking about, like where if you drop a mouse down a mine shaft, it'll walk away. A human will break every bone in the body, and a horse splashes. That kind of
Yes. Yes.
thing. And the same thing goes. The same thing sort of goes with drugs. I mean, they say that, but again, there are all sorts of individual things that happen within animals. So I don't know if you've heard about Tusco the elephant. So
No.
ages ago, I don't know. They're trying to like deal with elephants. They go into like this heat, this like mating like frenzy, and so they're trying to deal with that by
Mhm.
treating this elephant with LSD. Um,
Okay.
the problem is that no one had ever dosed an elephant before, so they assumed, okay, let's take a smaller animal and scale up the dose. So they said, like,
Mhm.
okay, here's a dose of a cat, and
Mhm.
and they use that dose on the elephant,
Yep.
but
Correlating their
yeah,
weights, right? Like the amount of times the cat is less than a elephant, that's the amount of times that you want to multiply the drug dose that you're giving to the elephant.
yeah. And
Okay.
what happened was that Tusco lost his rag and went really like kind of was freaking out a lot, and the trainers panicked as
Sure.
well, and they ended up like
Mhm.
dosing Tusco with a bunch of they and ended up with Tusco unfortunately dying. But yeah, so it's just because sometimes doses don't happen work the same for different animals. I mean, like for instance, like
Yeah, I mean
yeah,
metabolism is the rates of metabolism are different, right, in different animals. And then you also have to remember like it's also about like what the target of those of that intervention is, right? So like in the example of LSD, it's like, oh, that's some brain
mm,
receptors, right? But do you really know the difference in the amount of those particular receptors between humans, cats, and elephants, right? Question mark.
yeah.
So yeah, it's not as straightforward, I guess, right, as giving a dose that is the same, or even scaling down based on body weight. There are always like kind of caveats that can be built
Yeah,
in.
that's why in a lot of animal studies you see some very boring experiments up front to basically do this thing called characterizing the model, where they try to. So in this case, it would be like trying to find the lowest possible dose until it doesn't work. Uh, and I
Hmm.
feel like 1 microgram dose is kind of in that area where they're trying to get to a very low dose that doesn't work, and 5 micrograms is like the minimum dose that works, and then after that you kind of topped out and you can't really get any better.
Yeah, 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 absolutely.
So yeah, I took a long time to explain that. Like through LSD elephants. I mean, that was. I just like that story. I don't know why.
It's an interesting story. I mean, it's I got the image when you said a horse splashes when you throw it down a mine shaft. But top it with an LSD elephant story,
Yeah,
please. Go
definitely.
ahead. Go
Yeah.
ahead.
Kids who are coming here to learn.
um,
Yes.
moving on into Figure 1D, I guess maybe the most important antibody that we would be looking for is a neutralizing antibody,
Mm.
right? Those are the antibodies that actually prevent the virus from fusing to cells. And you can see that in 3 and 6 weeks post vaccination, you get these neutralizing antibody titers. And this is actually a nice confirmation that the boost dose is very
Mm.
important for this phenomena, right? Because without the boost, there's actually a bunch of people. There's a bunch of people. There's a bunch of hamsters that don't get neutralizing titer 6 weeks post vaccination.
Yeah. For sure. I mean, is it? I'm getting a bit of echo on your end.
Oh, really?
Yeah. Sorry. They also compare it to human
Okay.
convalescent serum. So that's in the black triangles. Again, you see like this interesting thing where the vaccine seems to produce more neutralizing antibodies than human convalescents.
Mm-hmm.
Uh,
How's that? Does it sound a little bit better?
uh, yeah, I think it sounds a bit better. Yeah, that's better. Anyway, so yeah, that's important thing to know. And also, like, I don't. It does seem like the 1 microgram dose is actually does quite a lot better on neutralizing for the prime dose.
Mm-hmm. Mm-hmm. I mean, right? I think this is one of those things where what is the effect of the LNP itself,
Mm,
right, on this immunity phenomena? I mean, we're not going to get to that answer anyway in this paper. It's something that I am very curious
mm.
about. But yeah, I find that very interesting that the 1 microgram dose seems to do so well here. And my one hypothesis I would throw out there is it might be something about the LNP, right? Like flooding the system with this compound, right? Maybe isn't the right way to treat the immune system.
Yeah, I mean, or you could have just had a group of hamsters who were just like fed better suddenly, or they didn't. I don't know. They didn't have much liquid in delivery. I mean, there's all sorts of different
Sure,
things that
yeah,
can.
it could be an experimental, right? It could be a systematic experimental error where in these three groups, like, yeah, they got treated differently.
But I have an idea about like perhaps. I mean, it would be quite interesting to delve into the differences between these dose groups because if there is a difference, then it'd be quite interesting to see like kind of the underlying factors for that.
Sure. But as I said, we're not going to get at any of that information in this paper because they end up only working with one of these groups
Yeah.
in their single cell,
So
yeah, in their single cell data set.
that's going to be a bit of disappointing, but we're going to get a lot of information anyway. So
Yeah. Well, let's plow
yeah,
forward.
let's
In figure S1 A and B, they take the spike specific titers and the receptor binding domain specific titers, and they show that it correlates with the amount of neutralizing titer that you get.
yeah.
And the correlation is stronger, right? In the more time you wait, right? And um, also between it's stronger in the RBD group. Yeah, it's stronger in the
Yeah.
RBD group, which you would expect actually, right? Because neutralizing antibodies should be binding to an important part of the spike protein, and receptor binding domain is an important part of the spike protein.
Yeah, so after week 6, you kind of see the. I mean, because it's hard to see the correlation in the early kind of stages. So on the first
Mm-hmm.
week, it's all very compressed because everything in the x-axis is to do with the actual size of the neutralizing antibody titer. So that's been quite small before the first like dose, and then as we see, it gets boosted afterwards. So because that we get that more variation, actually get to see that correlation a lot more easier because we get that.
Sure.
Yeah. So yeah, so that's
I mean, that's just what you're assuming, but I agree with you, right? Like, probably all that stuff that's clustered on the x-axis is before vaccination.
uh
Is only the 6 week, right?
yeah, so on week 3, like on our x-axis, it goes between an average of 1 to 3, and then on our like on next week it goes all the way up to 4. So there's like a
Mm-hmm.
whole extra. So what I'm hoping is that if you zoom in, then you'd see that correlation a lot more clear. But because they've zoomed out to keep them both in the same kind of region of view,
Mm-hmm.
they look slightly different.
Ah, yes, yes, yes. I see what you're saying. I mean, I think the way that they made their statement, I always just actually look at the number, the R number,
Yeah.
and say how close is it to one, right? The closer it is to one is the better correlation.
Yeah, I mean, there's a lot of other things you can do with regressions because, like, looking at where the correlation stops working can actually tell you whether there's a different pattern in happening. So sometimes if there's systematically more things, and you don't really get that from the correlation statistics, but it's
Sure.
so I've just ended up looking at both. But look, so yeah, there's lots of fun statistical stuff. So yeah, weight is what's happening next. So weight is something that all animal houses, anybody who keeps animals, uses that as an easy way to check whether they're healthy or not. So
Hmm.
if because usually weight is a kind of expression of sickness behavior. So animals when they get sick, they stop drinking, they stop eating. So it's something you have to
Hmm.
watch very carefully in your experiments because if so because you want to make sure that if like the animals are sick, it's because of the disease, not because they're behaving in a way that's starving themselves or something like that. So it's
Totally.
so yeah, weight is a very good like way of measuring that. So
Hmm.
there's a lot of things that can. So it's
Oh wait, it sounds like, but actually, from what you just said, it sounded like weight is open to one caveat,
yeah
and that is there's the like it's unclear whether it's specifically because of the disease or the behavior that is impacting their disease state,
yeah so
impacted by the disease state.
yeah
Yeah,
the
you
interact
can't separate them.
yeah you can't really separate them I think because I mean there is that physiological response that feeds into the behavior, but then like,
Mhm.
say if you're doing like a nutritional study where you're force feeding animals like a supplement or something like that, then you can actually end up making more animals survive on the supplement than in the other group just because you're feeding them, and so that means
Sure,
that
sure,
a lot of
sure.
supplements can end up looking like they cure a disease when actually all they're doing is you're feeding the animal, so you're allowing it to survive more, whereas in your control. So that's but
But
that's
they're not force feeding in this
no,
condition.
sorry.
There's like because the intervention is just the vaccination.
Yeah,
So, but yeah, it's a gross measure, right, of the course of a disease, and it can't be separated in terms of like the specific mechanism of why they're losing this weight. It's just like sick animals lose weight,
yeah,
whether it's because the disease itself is changing something in their metabolism to use up energy, right? Or they're just not feeding themselves properly during this
yeah.
time because they're so so symptom
Things
laden.
like sweating, frequent urination, all those sorts of things can have an effect. I promise I'll try to do less tangents, Danny, because I realize I have taken you on at least three different tangents already.
Totally, we're being very tangential.
Uh,
Okay, so let's push into S2 and S3. Here we have animals.
so
We're just looking at their maximum weight
yeah,
loss. You can plot them down, and you can see that like when you just take that, there's a clear difference between the mock infected group and the vaccine groups,
yes,
and you can draw a correlation between the max weight loss and their neutralizing titer. To say that neutralizing titer is helping, right, like animals not lose so much weight.
yeah, definitely. I think we can move on from that, and the next one is looking at viral loads.
Yes. Yeah, so looking at viral loads by two methods. One, they put in the supplement because it's not as accurate,
Mm,
right? Is the subgenomic RNA, right? Because subgenomic RNA, that's just the evidence that the virus is around. Doesn't really mean that there's productive infectious particles. So that in B, that's when they're looking at PFU. Those are actual plaque forming units,
mm.
right? Viral particles that can mount a successful infection and kill a small circle of cells on your plate. And yeah, basically at 4 days post-infection, there's not a lot of active viral particles being made anymore in the vaccinated groups.
Yeah. So like at day two, it seems that there's some differences between the groups, but there's like still viruses there. So but then it seems like the
Hmm.
vaccinated groups just clear it out a lot quicker, and they measure it in
Yeah.
two main organs: the lungs, which you're familiar with, and nasal turbinates. So everyone's familiar with like this part of the nose, but actually that goes all the way back to the back of the mouth. So the bit between like the
Hmm.
front and the back of the mouth, that's where your nasal turbinates live, and that's where a lot of
Hmm.
bacterial viral infections like can survive and give you colds and stuff. Um,
Yeah. because it's a big internal surface basically,
Yeah,
right? Like so like air comes in and you're trying to filter stuff out, but stuff will settle on that mucous membrane that is your turbinates. Yeah.
yeah, and like even when you're eating, all the food vapors go from the back of your throat into you. So that's how you can also have a lot of sensory kind of organs in that area that get your sense of
Mm-hmm.
smell. Um,
Mm-hmm. I guess the other thing to point out here is that at 4 days post-infection, they're still able to detect the subgenomic
mm,
RNA, right? So there's a disconnect here, right? That like there might not be any more infectious virus, but maybe the virus is still replicating inside the cells and it's just being prevented, right, from getting out. That could be something that's happening.
yeah,
This could be, right? This is the immune. To me, this is a nice illustration of like the immunity at
mm,
work, right? Like whatever the immune system is doing, it's doing it at this time point,
yeah.
right? Because like the virus hasn't stopped getting like replicating itself, making more copies. It's just that it doesn't actually make any. Yeah, it's not able to pop out anymore. Or it could be that the virus that you recover is already covered in antibodies or something like that, which make it unable to fuse. That could be
I
something.
mean, yeah, how I imagine it's
Um,
almost like a steady state where like the cells still have the virus and they're still like making it, but the immune system is just picking them off before they become productive. Uh,
yeah,
so,
yeah,
so
I definitely think that's something that's at play. Can't say exactly what it is, but it's an important time point. I think, like if they were to do other studies, which they don't, but like for other people who might be picking up this data, I think that using this model, there's an opportunity to understand something at this time point.
yeah,
Okay, so figure 3 BC, they do some correlations between the viral load, either measured by PFU or QRT-PCR, and the antibody titer. And as you would expect, it's a negative correlation, right? The more antibody titer you have, the less virus you have
mm, yeah,
by either of these methods.
yeah. So there's not really much more to say to that. That's what you want to see.
Yep, absolutely. Okay, another supplemental figure here. Here they're actually looking at different antigens, right?
Right, yeah. So they are looking at the the N protein.
Oh, well, this is just S
S,
for now. I guess maybe the next.
yeah,
Oh yeah, in the next one I have NP. So
yeah,
this first they just look at S. So this is, I mean, this is actually not. This is what we expect, right? We vaccinate with spike specific, and so we're getting spike specific titers.
yeah. They
Okay,
compare the baseline with the actual level of antibodies they have. So the interesting thing,
after they get the
yeah.
infection, yeah.
So in the final time point, you actually see the naive group that didn't get a vaccine have an antibody response, and you can also see like the difference in the baseline between the vaccinated and before and after. So you can even see that even the vaccinated group have more antibodies.
Yes, yeah, which really says it's working, right? Like the regular infection is able to produce that jump in antibodies because as we would expect, but it people that or people hamsters that are vaccinated, right, have that increased boost.
Yeah, and they also look at nuclear protein, which is uh, and it's the
Hmm.
reaction.
Yeah, so I like this is I find very interesting. So I think the time point they have here is the 4 day, right? Because for nuclear protein they only measured it once.
Mm,
I think that's the 4 day post-infection one. It could
yeah,
be the 14. I don't recall. Oh no, it's
14.
14. It's
Yeah.
14 days. 14.
So
Um, so actually, I like this, right? Because it's quite interesting from the standpoint of if there is a bigger boost with if you got vaccinated,
mm,
right? Because this is an antigen that you're not vaccinated for, but there's still a greater increase in some cases, right? Like for example, C. I think C is the best example of this, right? Where 2 days post-infection, there's like it's pretty similar, right? 4 days post-infection, you can start to see the gap emerging, right? And then 14 days post-infection, there's actually quite a big gap
mm,
between the antibodies made um, just after you've been vaccinated from S and after you were infected.
yeah. Uh,
So like just I guess the point here is just because you've been vaccinated for spike, it also improves your antibody response to other antigens.
yeah. I mean, the interesting thing is like seeing how diverse a prime because I mean I've been looking at the single prime boost one throughout this and how It hasn't
Mhm.
been. It's not so much that it has been doing badly. Some individuals in that group do go well, but some don't, and that's been interesting. Like that's why
Mhm.
you want to see like lots of individuals in a sample, because then you actually see that variance, and you wouldn't get that if you just looked at a
For
few.
sure.
Yeah.
For sure. And that mirrors though the data that we saw earlier, right? Like without the prime boost, the neutralizing antibody titer just wasn't
Mm.
high, right? It didn't bump. Um, oh yeah, right. C is not like a specific thing. C is neutralizing titer.
Yeah.
I was thinking C was NP, but NP they only give us one data point, which is 14 days post-infection.
And there doesn't seem to be any differences between the vaccinated and the non-vaccinated group for that, which is kind of understandable. I
Yeah.
think. I think that makes sense to me. Uh,
hmm hmm
but
Yeah, sorry, scratch that. What I said before, right? Like earlier when I was excited about C, that's not what people should be taking from this. Because that's just neutralizing antibody titer. That is spike protein,
yeah,
right?
that
Essentially.
is.
Yeah, that is spike protein essentially.
Yeah, and that's the thing that we really want to, because we don't. It's not just about whether the antibodies stick to it; it's about whether they actually stop the virus. And that's what neutralization gives us. Um,
Right. Right.
yeah, and now we start getting to the microscopy. So this is where they take slices of lung and put under a microscope. Um,
hmm
and so this is always takes me a bit of time to figure out, because studying these images is kind of an art, because they look like blobs under a microscope. It's hard to figure out the
Yeah.
individual organs. So you've got.
And later on in this paper, they will give us a table that summarizes their findings. But like, I guess we can take a moment now to at least try to see what a histologist would see.
So yeah, you've got the lungs, you've got the structural lungs, you've got these alveoli, which are like kind of holes that hold up the oxygen. So when you breathe in, that's where all the oxygen goes. And then
Mhm.
you've got these much bigger kind of tubes you see in there. Those are the bronchioles. Um,
Yep.
and in between them,
And
you'll see red patches. Those red patches represent capillaries that are carrying the blood, and the inside is the blood, so that's the red bit. Um,
mhm. Yeah, and the dark purple stuff, those are staining
mm,
nuclei. And so, all cells have nuclei, but some cells have more than others. And if you get concentrations of cells, that would also make like a concentration of purple, right? So you
yeah.
can kind of see from these outlooks like where all the cells are gathering. And that's what they pointed out with the star
Mm,
symbols, is that they pointed us to, oh, look at these regions that are extra purple, right? Like for histologists, when they looked at that, that's a sign that cells are moving to that area, which is part of the inflammatory process.
yeah.
And so it's part of the pathology of a lung, because normally you don't have cells like infiltrating in that concentration in the lung.
Yeah, and in the lighter group, they've got antibodies that can stick to SARS, so you can actually see where the
hmm
SARS is in these lungs. And actually, they take like congruent slides. So what you do is when you stain, you
hmm
take one part and stain it with H&E, and the other part you stain with the antibody. So they're actually looking at the same
Yep.
areas. Uh,
Yeah, and they can kind of show that the places that they're seeing concentration of virus in brown matches up a little bit to those places where the cellular infiltrate is happening. So, right in the locations with infection is where you're getting these inflammatory responses.
yeah,
And then, of course, really at the big level, you can see that when you're not vaccinated, you get tons more viral replication all over the lungs.
Yeah. So I mean, with histopathology images, like you can take a lot of information about like how much like damage there is just looking at them. You can say, oh, there's clear damage, but actually, what you really want to look is like the kind of damage that's happening. What kind of
hmm
what kind of cells are infiltrating? What kind of how they so uh, like lots of things called interstitial macrophages, like I think infiltrating in these sorts of areas. So like non-specific like
Sure.
phagocytes, I think are in there. I can't really see that. I'm
Yeah.
literally pulling that from the. It's very hard for me to spot them.
Are you and those things can, I guess I'm not really familiar. Can those things be separated just by H&E stain?
It's difficult because I think immune cells tend to have like much bigger much bigger nuclei, and actually if you look at them, if you're kind of trained, you can see the differences between the different types of immune cells, because like some have like eosinophils have like three nuclei that are like stuck together and it looks
I
very
see.
weird. Others have like more and
Mhm.
sometimes you can see like granules in there. So granules have all these like kind of chemicals that the immune system uses to kill bacteria or
Yeah.
viruses. So
Yeah.
like that's why.
Yeah, no, I I just wasn't sure if people typically collect that data with H&E, because I would think that a better way to get at that would be to stain for some sort of marker of that cell that you're interested in to then show that it's infiltrating in that
I
area.
mean, you are right about that, but I think with a lot of experienced pathologists, so this is like over 100 years. They've like named, so they named like cells after how they look under the microscope. So they go, oh, we see
Right,
granulocytes. So they're the ones that have lots of grains in them. We see like a couple of,
right.
so they. So I remember I worked on a paper with one like pathologist who would literally look at stuff and like pick out individual cells, and I think it's a lot of expertise and a lot of art to like kind of recognize them. It's almost like yeah, bird spotting or something like very one of those things where you sit and you like are an
Yeah,
expert in identifying that, and I just don't have the eye
yeah.
for that at the moment. But
Yeah, yeah. But it could be in these images. I would also think that to do that, you would also need control over the image, though,
mm.
right? Like you would need to be able to zoom in when, if
Oh
required,
yeah,
right? And you might not have that control from a static image as presented here. So, like a histologist who was working with this group, right, could have gathered that data for them. But if they didn't, then we can't really go back to these images and gather that data.
yeah.
Um,
They
yeah.
usually have like a whole slide. This is just a small section of a much bigger slide that they kind of take a bunch of like views of it and assess each part. And yeah,
Yeah.
I mean, you can probably get an entire computer to scan the whole thing in high detail, but that's uh,
Hmm.
a lot of money, and it takes a lot of time for people to individually. And I'm not sure they have the
Yeah.
technology yet to recognize these things using a computer.
Yeah. Yeah. Well, let's keep going. I mean, they give us more sections in figure S5, right? These are exactly the same, like these are very much the same types of images that we saw earlier, just in another location.
Yeah.
So you can see that even in the prime boost one, like they have some images, right, that have very dark. And like that's the other thing about images, right, is that like they're all just
Mm.
representative. So like you kind of have to take cues from what they're showing you. You don't really know. Figure S6, they're looking between, again, more of these similar images. They don't have the IHC
Mm.
anymore, but instead they have 2 days post infection and 14 days post infection.
Yeah.
And here they actually the body of the text did bring some observations to the table. They were saying like, um, that like the infiltrate location was changing between 2 days and 14 days post infection, that like I guess maybe they thought it like infiltrated in different spaces. Once again, I can't really tell from these things, so we can just flip through the images.
Yeah, I mean, the one thing so I think the bronchioles are a lot more easy to because they got this purple border around them that makes them a bit easier to
Hmm.
stick out. And some infections
Hmm.
that border is like the cilia, I think. I mean, correct me in the comments if I'm wrong, but like sometimes that
Yeah.
cilia gets eroded during an infection because a lot of inflammatory cells just have to punch through that to get to the area of infection. And so like
Mm. Mm.
so early on it seems like it's quite thin, and then when things are healthy, it's nice and thick and more purple looking. Um,
Yeah.
but yeah, and also I think what they're looking for is like areas that would be alveoli or like, and but instead it's filled with like these kind of blue-looking inflammatory cells.
Yeah. Yeah. And they keep going
Yeah,
because this image is really long. So like they have the 5 and the 1 in the
yeah,
next. But again, for those of us at home and us, right, who can't really read these that easily, right, they do summarize what they saw, right, what the histologists reported when they looked at these slides in a nice table in table S2.
yeah. I mean, one thing I'd
Um,
point out, like I think that viewers can probably see, is those red patches represent capillaries. So
mm,
you can actually see that between the 2 days and the 14 days, there are just more red of those red patches in the.
mhm.
So that's something that even I, a non like histopathologist expert, can kind of see. But
Yeah, in the 5 prime boost, it's like quite evident,
yeah,
right?
um,
I would say in the 1 prime boost, it's really hard to see.
mm,
If we go back to the 25 prime boost, I would say, yeah, I can definitely see it in the 25 as well.
yeah. So
But then once you get in the mock vaccinated, when you're competing that red against the
yeah,
purple
it's very
of
hard
the
to
cellular
see.
infiltrate, that's really hard to see. Yeah.
Yeah, I mean, I think that's because part of the inflammatory infiltrate isn't just to fight the infection, but also to help repair afterwards. So 14 days, there would
Yeah, absolutely.
still be some hanging around, perhaps. Um,
Hmm.
But anyway, that's my
But yeah, no, that's a good point. Like they also said that the capillaries were expanded, the blood. This is all consistent, right, with
mm,
inflammation. And they even help correlate that to, or not correlate. They even help locate that, right? They have paravascular, parabronchular,
yeah.
to say that this is where it's happening in the airway or in these other spaces.
And they got an actual expert to look at these and give their assessment about the level of pathology. So usually
Hmm.
you want to keep your pathologists blinded so that you don't cue them in to like what that you want them to say. You want them to
Yeah,
be as kind of neutral as possible. And so when they look at like 2 days, they kind of see everything looks kind of the same. But then like the later days where we know the infection is still going, the mock vaccinated group is like quite inflammatory. There's quite a lot of damage happening
yeah.
there.
Hmm. Hmm. And again, that also correlates to just the amount of SARS-CoV-2 antigen that you're seeing by
Yeah.
IHC, right? So they're not unable to control the growth of that virus, and thus they are getting a lot of inflammation. Okay, so in figure S2A and S7A, they just want to point out that there's one animal that they have in figure in the five microgram boosted vaccinated group that didn't do so
No.
well uh, for some reason. And when they did their technique of single cell sorting and amplifying things, which I guess we're going to talk about next, that ended up separating from the group. So they have an outlier, basically, they wanted to draw their attention to.
Yeah.
We may have to flip back to the slide after we talk about
I
single cell.
mean, for sure. I mean, one thing I know is like when an animal loses about 20% of their weight due to weight loss, that is almost like a sign of oncoming fatality. So, for humane reasons,
Hmm.
they usually decide to cull them at that point, but to give them a more like humane way of to reduce their suffering rather than to let them suffer longer.
Hmm.
So that's why it's probably
Yep.
removed from this group. But they do this extra work. They do look into it, see like what was happening with that, because that's an unexpected result, and sometimes unexpected results can yield quite interesting results. So
Well, do you think that's why they chose to do all of the sampling at 4 days post-infection? Because of the one
I
thing?
mean, it wouldn't.
That would be very interesting if that was the choice. I don't think so. My guess is it just like I agree that like uh humane euthanization is important, right, for an animal that went down that path. But I would think that they had pre-specified 4 days
I
post-infection, and so like this animal just ended up getting culled on that
Yeah,
day.
I kind of think that I agree because it didn't actually reach the 20% kind of threshold, but it was
Yeah,
on its way there, and so there was something very odd
yeah,
happening with that animal. Um,
yeah.
But anyway, so now we get to the meat of the paper. So all the previous paper is like stuff that I'd have been doing 10 years ago in terms of vaccination studies. Uh,
Yeah, and it sets up our understanding of what is the disease that we're seeing, right? And how does the vaccine prevent it? And or sorry, does the vaccine prevent it, right? And it does, right? By all the measures that we know in the
mm,
past. But one of the highlights of this paper is this other technology.
yes, single-celled technology. So here's a brief explanation. So all cells use RNA to make proteins that help them to function, and we have all these different cells in our body that do different things, and they have this, and and these different RNAs so each cell kind of has a different makeup of different mRNAs. So let's say if we want to
Mhm.
like say just count them, we'll be able to almost get an idea, get a profile of what each cell is based on the mRNA that it produces, because we know that
Mhm.
we at least know what job each mRNA is doing, and so we can just figure out what
Mhm.
job the entire cell is doing based on which mRNAs it's producing. Um,
Mhm.
how do we actually measure this? How do you measure each single cell? In this study, they use this thing called a gel bead emulsion. So these are these weird liposomes. They each have this barcoded RNA in them. So it's an RNA with a
Mhm.
unique sequence that they kind of can pre-specify and they can identify. Uh, Um, so
So I think the bead is covered in RNAs. There's a
yes,
picture of this earlier, right? The bead is covered in RNAs, and they're going to put those beads in an emulsion.
yeah,
Yeah,
yeah.
which is emulsion for, I guess, very cooking term, right? At least that's how I think of it. It's the mayonnaise, right? It's when you're kind of getting droplets of oil suspended inside of a liquid,
Yeah, they're
a non-oil.
yeah. I mean, they're almost like shells. So I think almost like nanoparticles, like the same as vaccine, where they keep the inside separated from the outside and stop these barcode
Hmm.
RNAs from like mixing with each other. Or
That's right. Yeah. So the barcode RNAs are attached to beads that will sit inside, be separated.
yes,
But I think you have a better image coming up about that.
yeah. So what they do is they pass these beads through this like kind of single like pipe, and then they mix them with individual cells. So again, they pass cells through the same process to get them as single cells in a line, and they mix them so each bead gets one cell, and then
Yeah.
and once those cells are in there, they pop those cells open to release all their mRNAs, and those mRNAs end up binding to the barcode RNAs. So
Hmm.
so now you've got all these different RNAs, but they all tie to a single barcode, and and then like these lipid particles get popped open. You've got all these RNAs mixed together, and what happens is that
But because they are barcoded, we know that they came from single cells, right? That's kind of the
exactly.
important thing.
So when you put
And I just wanna make sure to add in that we used oil,
mm,
right, to separate those pools,
yes,
right? So like inside of the with the bead and the barcode, that's liquid,
mm,
right? It's because cells want to live in
yeah,
water, but they're being separated by oil, right? So this is like droplets of water being passed through like an oil tube.
yeah. So this
Okay,
it's
anyway.
almost like having 100 million like individual reactions that are all separated from each other. So
Yeah.
uh,
I've always wanted to get this toy to show people like when I teach classes or stuff, like because it's a really good toy that I think shows what's happening. Do you recall it's like it's like an hourglass, but it's filled with oil and water
right,
instead of sand. And when you turn it over, the oil drips and you get little beads of oil and they kind of like go down like a little maze path or something. And that's essentially what's happening here, but in reverse, that the oil is everywhere. Actually, no, usually those things that it's colored water
yeah.
and it's maybe a neutral oil, right? And the colored water gets dripped. That's essentially what is happening here, just at a very, very tiny level. And then imagine every one of those drips has a cell inside of them and some barcoded RNA.
Yeah. Oh, I think I know exactly what you're talking about, but it's so hard to describe. But because I remember I used to have
Yeah,
one of
yeah,
those toys
yeah.
when I was a kid. But yeah. So now we've got this pool of RNAs, and but each ones that come from a single cell have the same barcode, so you can actually work
Hmm.
back. So using that barcode, we can pick out all the
Hmm.
cells with the same barcode to pick out all the mRNAs, and then using those mRNAs, we can Sherlock Holmes deduct what cell that it came from,
Totally.
and that's what is used to build these giant maps of what cells are in each in the lung.
Yep. Yeah, absolutely. I mean, you can imagine that that's a very complicated data set that comes out, right? Like you're getting a huge list of
Mm.
RNAs, right? And you're saying each of these RNAs has some unique identifier. And that's like a big table, right? Like it's like a table that has many different rows. And the way they choose to show it is using this. And we've talked about this before, actually, in a previous episode. I think it was the neuropilin
Mm.
episode, because they also use single cell RNA-seq there, but they separate. They separate that out using a computer that is just it's an algorithm that is able to find good separation between very complicated data sets, right? So there's variance, right? Like of course, different cells are making different things in a way that doesn't need to identify markers at
Mm.
all, right? It can separate out these groups. You don't know what the groups are, right? In some ways, they're meaningless because it's just a computer saying that these things are different, so they should belong together. And that's what you get in these clusters. But you can see in this map, they actually got
Mm.
labeled, right? So the way they got labeled was through our understanding of cells. Like if we can move to figure S8.
Yep. So cell type specific markers. So now we know that some cells have specific markers, so we can actually search through all the cells that express that marker and be like, okay, well we know that's definitely a T cell. We know that's a helper cell, and
Exactly. Yeah, so they even label here nicely, right? The different markers and then the cell type they think that corresponds to. And you can see like T cells is a really broad category, right? CD, 3D.
mm,
And you can see many of the blobs light up for T cells.
Yeah.
right? Because it's a very broad category, right? There are specific types of T cells that you could get more specific in getting, and those will be separated by a different marker. Do they have that marker here? They do not. But like that marker would be CD8 or CD4, right? Is a great way to separate T cells. So you know that one of those blobs is probably CD8, one of those is maybe CD4. Um, and then even I guess NK cells light up uh, for that particular marker.
Yeah. So in the
So
previous one, they'd literally taken all their data from like vaccinated, unvaccinated, all that to create this map, which almost gives you the possibility of space of where each cell could potentially lie. Uh,
hmm
and
hmm
so this is all like kind of so this figure like so figure 5A is almost like a map to give you an idea of what to be looking out for, like where things will be. Um,
3A, figure
oh yeah,
3A is a
3A,
map.
yes.
Yeah.
And 3AB is splitting up that map into each section. So looking at
Yeah. So I pulled the map out just so we can see those labels again.
yeah,
But you split the data set into the three different groups, right? One, the naive group, they never got infected, they never got vaccine. One, the mock vaccinated, they didn't get vaccine, but they got infected. And then one, the vaccinated and infected, they got the vaccine and they were infected.
yeah, and we can almost see like almost instantly there's a massive difference. There's in interstitial macrophages which basically
Mhm.
only seem to appear in that mock vaccinated group. There's very few in the naive and normal vaccinated and the vaccinated group. Um,
hmm hmm
uh, I mean, other small differences picking out. There's some differences in NK cells, some differences in CD8 and CD4 T cells. There's
Yeah, it looks a little bit less.
lots of granule
In
cells.
the vaccinated group, there are. Oh, I guess these have to fit into the map. I feel like there's a lot of changes in the bronchial epithelial
Hmm.
cells. They don't actually talk about those in this paper, right? But like you can kind of see that those green dots look quite different in the mock vaccinated group than in the naive and the vaccinated infected group.
Yeah, it'd be interesting to think about like the because it could be the damage to the virus, could be damage due to immune overreaction because those cells are just
Mhm.
sitting there and things are happening to them. Really, you don't. Well, actually, that's how I think of them, but I could be wrong about that. They could be doing something here, and that's an interesting
Yeah,
thing about this kind of study.
granular sites, right? You said that those are the purple.
Yep.
They're like there's a lot more of them in the mock vaccinated and infected group.
Yeah, and that kind of ties into the histology we saw earlier as well. And this kind of brings out,
Yeah,
but we get so much more detail here that is really useful.
absolutely. I mean, I think that's the reason why this is the star technique of the
Mm.
paper and like sort of their primary contribution into it. It's like they have this really well characterized model, right, where you can look at all these metrics that are easier to collect even, right, and they correlate well with disease states that we see in
Mm.
humans, right, or in other animal models. But then they can back that up and dive deeper into that those same animals and say what are the cell types that are going up and down. And again, this UMAP is one way to show that information, right? Where you're sort of asking a machine to cluster things. But once you know, if you knew the markers, if they know the markers that are going into making a cell type, they can just extract that from the table. You don't have to do that visually,
Mm.
right? You can just take your big row of all the things that are expressed in every cell and say, for all cells that express the markers of granular sites, right? How do those levels, right, increase or decrease between these three groups? And so that is what we're seeing in the next figure,
Yeah.
3C, right? Is that it's the same data, right? Like UMAP was just one way of showing it. UMAP is a way of showing like that huge list of RNAs, right? And here they just go in and they extract from that list and they show it into us in bar graph form, so you can more clearly see the abundance differences between these different groups.
Yeah, the thing that sticks out to me here is how, like, in the naive group where there isn't an infection, you see a certain number of like CD8 T cells and CD4 T cells, but and same in the vaccinated group, but in the kind of mock infected where there is no, it's they're much lower, and
Yeah, yeah,
so
they are much lower.
I mean that. So usually I'd say that I'd have expected like say the C those the vaccinated and like both unvaccinated groups to be at the same level and the vaccinated group to be higher. But actually, it seems like here it seems to indicate that SARS is doing something to make the T cells like be expressed lower, and that's kind of interesting to me.
Yeah, yeah. Something is happening that impacts the population of those cells for sure.
I
Um,
mean, again, it ties back to that BCL6 paper we did a long, long time ago, where that
uh-huh,
showed like there's some T cell dysfunction happening during severe SARS-CoV-2 infection. Um,
totally.
so,
um, yeah, kind of interesting. They do have B
mm,
cells. They're not in this one, but if you go back to the UMAP, they do look at B cells. They're like an orange group
yeah,
that is just south
yeah,
southeast of the CD4 T
it
cell
looks
group.
kind of like a sailing ship.
Yeah, it is a little bit. It does actually seem like they're
Mm,
decreased, but they don't show that actually in our. Uh, they don't show that they didn't pull that out, and I think this is where you really get to what do you do after you've done this
mm,
experiment, right? Like you've done this experimental design, we know what it can show us, but then how do you start explaining this to people? How do you interpret the data that's coming out to like see the differences? Obviously this paper has a reading, right, that they're trying to draw out by showing us very specific comparisons, but any person who is trained in immunology, which we are not, like, at least not like in the depth that you would need to start making your own hypotheses using this data, could go in and look at this data set and try to pull out a story that they think is happening, right? Based on these different fluctuating numbers of cells, right? How is the immune system responding? Of course, we can trust the experts and say, tell us, these people who did the experiment, but there are other experts out there. There are other
mm,
peers, and this is a preprint, right? This is something that hasn't gone through a review process yet. I actually believe that the story that they tell us here will be modified somewhat when we see this in its final published
mm.
form, because it's going to rely a lot on those other experts around the world in immunology put feedbacking into this data set and saying, hey, did you think about this?
Yeah,
Like, it connects to this piece of literature that I'm an expert in, right? Like you should bring this story in and say that your data can support this or doesn't support this, right? Because all of that information is useful to understanding how, right? This is the data set of how, but like the actual explanation of it is going to be very reliant on lots of different experts with different pools of expertise.
yeah, and they have all this data, so a reviewer can
Mhm.
ask them the most obscure question, they can pull out like an answer to that.
Exactly. Like the expert in like plasmacytoid DCs that express the marker CD, 169. I mean, I'm just pulling that out of my ass, right? But like, you can name any marker, right? Any obscure set of markers that define a specific cell type, and ask the authors of this paper, ask the data set, right? Is it changed, right? Does it change? And if it does, right, then you can start imagining the hypothesis that supports that, right? And then maybe designing some follow-up experiments to show that this is a phenomena that happens, especially if the subset you're defining has been found in
Mm.
humans, right? That would be amazing, right? Like if it's really well known, the subset that correlates it to humans, then those would be, in my opinion, the most important follow-up studies that come from something like this.
Yeah. So next one we going back to the outlier.
Back to the outlier. Yeah, back to the outlier. So originally, when they defined the outlier, they basically just instead of using UMAP, they used PCA, and they just looked at the bulk RNA and said, even when you don't divide it by single cell type, it's just really
Mm.
different. Like the RNA just like is really different. But here you can see it on the UMAP, and you can see that that RNA is very specific for granular sites. They have a lot more. Yeah, they have a lot more things. Sorry, that's my phone. um, they have a lot. They have a lot more reads, right? In the granular site area, and when you segregate the bulk RNA um, in the by granular sites again, even it's very different. Like it's not the same granular sites that are existing in this outlier
Mm.
sample that exist in the other sample. So there's both a whole bunch more of them, and they're expressing very different things than all the other ones.
Yeah. So if you look at that graph, there's a whole new island just for this outlier. So like on
Yeah.
top of they've got our normal map, and the bottom you've got this entire island of abnormal granular sites. Um,
Yes.
so they do delve into a bit more about what those things do by looking. They've got this volcano plot. Uh,
Mm-hmm.
which essentially, like, on the bottom axis is kind of the effect size of the differences of different genes. So like the further away, the
Yep.
bigger like the change in the gene. So going down, and up, and the actual,
Zeros in the
yeah,
middle.
zeros in the middle, and kind of the y-axis is on the significance. So how kind of certain we
Mhm.
are that there is actually a difference. So you'd want to see like
Yep.
a U on things that are like on that par of significance.
Yeah, absolutely. Yeah, so you kind of start reading it from the two branches and then you move your way down into the center. And things that are too close to the center, they're not that
Hmm,
significant, so you're more people generally just don't interpret anything in there, right? It's
right.
like it's too insignificant and too small of a change to matter. You just like interpret the stuff on the two branches, and they can take the stuff that they find on the branches and say like, what are those genes used for? What pathways do they sit in? Right, and then they can just say, oh, here are the pathways that they sit in. That's D, right? Upregulated and downregulated pathways. So all the genes that clustered on the left side of the graph, that's going into the downregulated pathways. All the genes that cluster on the left side of the graph, that's going to upregulated pathways. And this is also a way to start telling a story, right, about what the cells are doing, right? These cells, they have upregulated neutrophil degranulation and activation and proliferation, but they have a downregulation of suppressive responses. So these are the granular sites that are getting into the
Hmm.
lesion at the very beginning, right? They're the ones that are infiltrating and saying like, let's destroy things. That's what's happening. And if you read the text of the paper, they end up saying, well, I guess that's kind of consistent with what we saw because they only sampled like one lobe,
Hmm.
I guess, of the lung to go into their single cell RNA sorting. And they were like, oh, that lobe that we sampled, it did seem like it was extra inflamed. And maybe like we were sampling like a place that had like an active like just a huge inflammatory cluster of cells happened to be concentrated in the lobe that we sampled.
I mean, yeah, that's the. I mean, I've seen this as something that the reviews would jump on immediately because okay, they're basing this hypothesis on histopathology that they're not showing, and they
Yes.
and they kind of are trying to say that it's a heterophilic cellular lesion, which they don't really talk about. But heterophils are this cell type that is only found in like certain animals. Like they're found in guinea pigs, they're found in birds and reptiles. They look kind of like well, they behave sort of like neutrophils, but they look like a different cell type, so they're called heterophils. So they,
Mm-hmm.
so I think this is another instance where like the hamster immune system is slightly different. They're almost trying to like use it as an explanation, like oh, there's some kind of difference in the hamster immune system that is causing for this heterophil cellular lesion that is unusual, and
Mm-hmm.
um it's I mean
It might not be, right?
yeah
That might not be the case,
it,
right? Like that to explain it away just based on the model, like that might not be the right approach, right? And I think what you're saying is it would be nice to see more data, right, around this in order to just fully exclude it, right, and not say like maybe the sample size was too small, and if they scaled it up, they would find a bunch of hamsters that went down this path.
I mean, I think a lot of it is the problem is that hamsters as a model haven't been studied very well. They haven't got as much attention as mice, so there's a lot of stuff that is mysterious. So there could be something, and they. So I feel like if we learn more about hamsters, we'll be able to go back to this and look at all the how these kind of uh, things are happening. But the thing is, with outliers, it's always difficult because you don't expect them, so you don't know the right way to analyze them. So it's hard to
Yes.
like judge the whole paper on them because actually getting this amount of information from an outlier is very good. I mean, usually for some places they just get rid of the outliers and they don't tell you, or they just try
Mm-hmm.
to just tell you
Mm-hmm.
it's an outlier and don't worry about it. With this one, they actually put that effort in to try and provide an explanation. I feel like
Yeah.
that deserves some credit,
Yeah.
but
to
yeah,
me to me I think it's important because the animal was the more valuable resource in all
mm,
of this, right? Like because they went through this process of infecting an animal, right? Like and they might as well continue running that sample, right, through all their analysis pipeline, which is the
yeah,
whole single cell RNA seq. So yeah, I mean, again, I do applaud them for showing it to us because I think that that's a very transparent practice, right, for the data. But yeah, I don't know. I mean, to me, it's just it is a strange thing. I don't know whether or not I didn't feel like from reading their paper, I felt super okay. That's why we should call off this, right? Not think about this
mm,
outlier at all. The other thing I'll say is it also highlights the fact that Single-cell RNA-seq is only as good as the sample that you
Mm.
put into it, right? So if they were dissecting the lungs in a certain, like, it all matters, right? Like, the tissue looked weird. If the tissue looked weird and they put it in, right, and it gave them a strange result, right, like that is actually, that impacts things, right? Because the lung is a big place, and so they don't put the full lung into the sample.
Mm.
They only put a piece of the lung, right? Maybe if they put the full lung in, it would have averaged out this thing. That's what I thought they were trying to say. That,
Mm.
like, that it would have averaged out if they put the full organ sample in, right? And that could also be a reason why you would discount this outlier, right? Because it is really just a sampling error on their end.
Yeah, I mean,
Yeah,
it's the thing is, like, things like this, it might be just something that's bizarre and never happens again. But then if it happens again in another study, then we can go back and find out that there's something interesting going on. So
absolutely,
I, I recall
absolutely.
that, like, when I was doing a PhD, I had something similar like this happen, and uh, at first it was like discounting it, but then it happened again, and then it turned out to be something quite interesting. That
Mm-hmm.
so it's
Mm-hmm.
so that sometimes, like, things like this, it's something you can go back to if there's something and investigate more. It's a hypothesis generating, because I think the most interesting thing about science is when you find something you don't expect, you can follow that. I think that's what they kind of did here. They tried to follow it down, and yeah, it's interesting, like, how, I mean, this entire, like, island is like that they find in of, like, granulocytes is interesting, and learning more about
Mm-hmm.
heterophils, what they do, it's there's a lot of opportunities here with outliers. Um,
Okay, so let's continue on, because now they're gonna just show us a bit more about their main data set. So they've excluded the
mm,
outlier, right, for the rest of this paper, because it's strange. They're trying to say that it's specific for something else, but they're not going to pay attention to it in the rest of this paper. Here they're going to look at viral gene reads, so just seeing, like, because they can also just align the reads to the viral genome and say, oh, look, we're getting virus to grow up in very specific cell
yeah,
types. Um, and as you expect, and there's really not a lot of viral reads in the vaccinated
mm,
group. It's mostly in the mock vaccinated infected group. And those things are clustering in macrophages and granulocytes.
yeah, and moving on, they actually, like, break it down by each individual in each animal group, so you can actually, and they also break it down by gene, so looking at which genes get expressed. So
Mm-hmm. Mm-hmm.
so one of the ORFs gets really ORF1AB gets seems to be expressed very highly. It,
Yeah, but ORF1AB, I think that's the polymerase,
yeah,
right?
you.
So, like, of course, like, if these cells are infected, then they have polymerase in them.
I mean, for me, the interesting thing is how is looking at the animal groups, because you can see, like, two animals have lots of that, but the other two
Yeah,
don't. Uh, so
yeah.
I think this is an interesting thing. Like, infections don't really average in the same way. So if you do it, an infection in one animal might take a different course to another. So it's very hard to actually
Hmm.
find out this platonic ideal of what an average of an infection is, which is why looking for the variance is kind of interesting, and looking at things that behave differently.
I think, once again, I also think that this also really matters, like, their sampling
Mm,
methodology, right? Like, some of these differences could just because they sampled something differently,
yeah,
right? Because, like, lesions don't appear, like, equally distributed throughout the lung, right? There's, like, random chance where, like, oh, more viruses went down your left side than your right
yeah,
side, and then more lesions were on the left side of your lung than the right side of your lung. There's chaos in an infection, right? In terms of initial conditions, even in the most controlled cases, right? You could inoculate with virus and things could go down the left lobe, but not the right lobe, meaning that the left side of your lung is going to be more inflamed than the right side. And those sampling differences will play out in these data sets.
mm,
So I feel very cautious about trying to interpret so much here, because because it seems like it could be, it could be very
yeah,
variable based on just the way the experiment was done. And I mean, I think that applies too to these different cell types as well.
mm,
You try to average it out, I think, by doing all these multiple samples. And I think in some cases it works, right? Because when you average this all out together, we do see some differences between the cell types that are present,
mm,
right? But when you zoom in closer, which is what is happening here by each individual animal, right? You start to see differences, and you're like, what does that mean? Well, I don't, I
Mm.
don't know what it means as much as anyone else, right? It might just be the variance of the sampling until you can control it or follow up or bring a hypothesis to the table, right? That helps you gain some clarity on it.
Right. So CCL8. So b- based on like previous work in ferrets, they looked for this gene specifically. Um, this is
Yeah, and they find that it is upregulated in the same cells that are infected, right? Those interstitial macrophages and the granulocytes, a whole bunch of other cells as well. um, and yeah, and so they think that that is why this is like it's a chemoattractant. It
mm,
brings other cells together, and that's consistent, right, with the way that we think infection goes down, right?
right.
Like that you have all this inflammatory infiltrate, and that's what's causing the immune system to go wild and maybe miss the important part that they have to suppress this particular virus infection. So they actually advance that as their favorite hypothesis,
Mm,
right? To the difference between vaccinated and unvaccinated in this sense is that when you're unvaccinated, you get this like crazy recruitment
mm,
of cells, and that's partially why those animals are not able to deal well with the infection.
right. And now we're going to Figure 5AB, where they do a lot of correlation stuff.
Yeah, and I think this is again, this is to get at the like they're trying to reinforce their favorite hypothesis, right? Is that this runaway immune thing, attracting all the cells here, is what is bad, right? Because a disease is can be as much a part of your response as it is the antagonism from the pathogen. And so they're going to, because they have so much data from the response side, right? They're going to compare, right, the two different responses, right, of the immune system just by looking at the genes that are being upregulated,
Mm,
and yeah, the differential expression pattern. So in some ways, it's like this is like a volcano plot versus a volcano plot, is
right.
what they've
Yeah.
made for us. Volcano versus volcano, and it's going to give you a sense of how much in each of the two cases things are changing. And because so the line, if the line goes whenever you're looking at big graphs like this, it's always nice to anchor yourself on something like the y equals x line, right? The diagonal,
mm,
right? So if the correlation lay on the diagonal, it would mean the responses are the same, right? The
mm,
genes that are lower regulated in the genes that are less expressed in the VI and MI group are the same as the genes that are expressed in the MI versus the VI group. Um, but you see this slant, right? That kind of pushes it
mm.
more, and that just means that the magnitude of the response is going to be higher for one versus the other. And I guess because it's being slanted downwards, that's closer to the mock infected group, right? And so it's the mock infected group that has a greater immune response. And you can see that too in the
Yeah.
Venn diagram, right? Is another way to see it is that there are more genes, right, that are uniquely on the Venn diagram side for mock infected than on the vaccinated. Um,
Yeah.
and that's their hypothesis, right? Like they're basically the next parade of graphs we can show is really just that, all stuff to emphasize this relationship that something in the mock infected animals, they're doing a lot of stuff. Their immune system is doing things.
Yeah, there's a lot of stuff going, and the vaccinated groups, their genes tend to be a lot more similar to the baseline, like the of like people of individuals with no infection. Um,
Not
yeah,
sorry, not more similar to base. Yeah, I guess more similar. Their genes
so,
are more similar to baseline, but also that they are also doing things in the mock infected group, just not as
mm,
much, perhaps,
yes,
right? Maybe the magnitude is smaller, right? In the mock
right,
infected group.
yeah. So that overlap of genes in the mock infected group, but those are the things those are genes that are quite important, I feel, to the
to
immune response.
yeah, to all immune responses, right, whether
Yeah.
vaccinated or not. And like, right, like there are things like in both vaccinated and mock infected group, you're going to have to, get cells to the location of the infection, right? Like it's just whether or not like that's appropriate. And I think this is a big concept in immunology that is very intuitive for people who work in immunology, but maybe not intuitive for people who aren't immunologists, right? Is that it's all about that balance, right?
Mm.
Like, and this does come out because people are crazy about inflammation. Inflammation is like
Mm.
such a broad term, right? Like, and sometimes you need a little bit of inflammation because like that suppresses, but too much inflammation, that's not good. And
Mm.
so that's basically what we're seeing here, right? Or that's the story that they're choosing to tell us here, and one that resonates well because it is a very well-worn concept in infectious disease, right? That like you want to hit the sweet spot, you want the right response, the response that's tailored for the infection that you're having, not one that is just like wildly trying all these different things that end up giving you a lot of symptoms that maybe don't clear the virus at the end of the day.
Yeah, so I'm gonna breeze through these like next few slides because they all kind of show the same thing but with
Yeah,
different cell types. So
yeah.
we've seen it
In
in
in all the different cell types, it's the same type of correlation, right? That line angle is dipped down, right?
mm,
And so it is. It's saying that mock infected response, there's more of it, right? It's at a higher magnitude, but ultimately there are a lot of similarities between the two because we're seeing a big set. I guess the plasmacytoid DCs are really quite different, right? They have
yeah.
very little
Mm.
genes in common between the response of mock vaccinated and vaccinated infected. So like that could be like something that you want to see follow up on, right? That would be a place that this paper would be telling the scientific community maybe we should be studying plasmacytoid disease in infection, and we may
Mm.
find right some key mechanistic factors that determine the difference between productive infection and non-productive infection.
Yeah, and
Yeah. Sorry, I said
the
infection. I
next
meant immune response.
yeah.
Yeah.
Next point. So it's interesting to look. So looking at gene by gene basis is one aspect of this, but I think they take the next step and look at networks as well,
Mhm.
because genes don't act individually; they act as part of a network of different functions, and so they kind of use a clever AI to put those together, and so you get these network diagrams where there is so using this thing called module-based functional enrichment analysis, which I'm not familiar with, but I'm name checking it so that you guys can put up in the comments and maybe explain it or
Yeah. Or
point
you
out
can
where.
Google that term, right? And you can try to find more information about it.
But the aim is to not just pick up genes, but pick up entire pathways that are being upregulated and
Mhm.
look at like how those what exactly is happening. Because looking at the whole pathway, you can actually get an idea of what the cells are actually doing and
Mhm.
how that's different between the vaccinated and unvaccinated.
Mhm. And so they have the common pathways. In the first round of like graphs that we'll see like this, they look at the common pathways first, right? So in my mind, these are not interesting, or
Mm.
these are things that right. This is again the common in the DC. This is in infection. These all have to happen,
Mm.
right? Like, and it makes sense, right? Like they have to do these things. It could be that they're not doing them at the right magnitude, right? In the different in the two different cases, vaccinated or unvaccinated, but they have to
Mm.
happen, right? The downregulation and upregulated things, they are part of the infection process. Uh, yeah,
Yeah.
I don't know. I
We
mean,
could.
like you could
Yeah.
you could go crazy in these things. Like we could tell stories about any one of these areas, and that's what I was saying at the beginning of launching into the single cell section. Is that it kind of matters like what you think as a scientist is happening in the infection, and of course that's based on the papers that you've read, right? So like we've read papers, so we would look at things and we'd say, oh, that makes sense based on this thing that we read, but you could get countered by somebody else who said, oh, but how do you explain this then? Right, is like kind of the
Yeah.
process of understanding this sort of data set. um
Yeah. Uh,
yeah.
we could like spend like the next hour going through each individual point. So we are actually trying to gloss over this. If you want to look at each individual point, you can read the paper. Link to the papers in the description if you want. but I think a lot of it is like very generic.
Yeah, so
Stuff that
yeah,
you
let's just talk about the categories, right? So they have these are
yeah
the ones in common. In the next one, S10A and S11A, we're looking at unique, right, for CD8s.
yes
And NK cells. Oh, sorry, unique in mock infected, right? So these are things that are when you don't get vaccinated, right? These responses happen. So this is interesting, right? These are like
mm,
non-vaccinated responses in infection. These may be things that the virus is tricking us to do, tricking the
yeah
body to do, right? That aren't productive. That's one hypothesis I think that you could generally throw out there.
so
And then we continue the list of things that are mock infected unique now in monocytes and DCs, conventional DCs. So again, things that the virus could be tricking us.
yeah
And then we have more PDCs. It continues. And then the final list is things that are unique in the vaccine infected or vaccine induced. And this could be stuff that tells us how the vaccine is working, right? So this is like, oh, maybe the vaccine is forcing us into these responses. These are very important. So there's kind of two ways at getting at, right? How to beat the virus, what the best immune response to the virus is. You could go from the side of what is the virus trying to get us to do, right? What is the virus trying to get the body to do versus what is the vaccine trying to get the body to do, right? And those are two ways of saying you don't want to avoid the things that the virus wants, and you want to you want to encourage the things the vaccine wants.
so yeah, the main thing that sticks out of the vaccine is like how little the differences are. It seems to be very subtle. So I think the key thing that we can take away is not so much the magnitude of the immune response, but quality matters.
Quality
So
matters, absolutely, yeah.
so
But that's true. But see, that's true of all infection, right? In some
mm,
ways, right? All infection quality matters because when you think about the ways in which pathogens, like just like broadly, right? Like I think I'd like to hear your opinion on this too. But like broadly, when you think about virulence factors, right, and immunomodulatory proteins made by pathogens, they tend to sow chaos, right? Like that's the like
right.
they tend to say, oh, like by the way, you should do this, you should respond in this way, right? Throwing up red flags and red herrings in ways that direct the immune response and split the strategy so that there's no concise way to deal with that particular pathogen. Is that would you say that that's right in your experience as well?
I mean, in my experience, but again, my experience isn't necessarily as massive as looking. I mean, there are so many different virulence factors out there, but I think that like yeah, if you get a disease, there is something that that bacteria or virus is doing to counter the immune system in order for it to get to the point where you notice it.
Mm-hmm.
Um,
So yeah, so I think that you're right. Like there are also virulence factors that work on very specific mechanisms that are important for their specific lifestyle, right? But if you think about how that would show up in a panel like this, it would
mm,
also show up in the same way.
yeah,
that that like the immune like because you'd be lighting up multiple immune pathways then, right? Versus the one immune pathway that is important to dealing with the particular infection.
yeah, I mean, I think it's interesting to like flip that and think about the counter. Like, imagining a bacteria that has no way of dealing with the immune system.
Mhm.
Then you'd see like a panel that looks very similar to the vaccinated one, where the immune system is just doing its daily business
Yeah.
rather than
Yes.
going out of control.
Yes. Yes.
So
Yeah, no, that's
that
a great. I love that. That's a really good way of interpreting this information as well. Yeah, totally.
yeah, so actually we don't want information, and a well-regulated immune system with a well-regulated response wouldn't go out of control. It'll just do each thing that we see big but smaller. So we
Mm-hmm. Mm-hmm. But life's not like that, right? Because
mm,
pathogens are also alive, or well, viruses. Okay, philosophical debate, but like they're doing things. They have some sort of intent, right, with the systems that they're engaging, and so you can't just have it doesn't it's not as easy as going from A to B because you have this counteracting force that's saying like, oh, actually maybe go down this path, or also just complicated mixtures are more difficult to deal with, right? That like it's gonna light up a lot of different pathways. Um, yeah, so as things get more complex, you would also just get more response as well because they're doing different things. Yeah.
yeah. So, like, overall, I thought that there's quite a lot. This paper covered quite a lot, and I feel like there's so much that can be pulled out of here.
Mm-hmm.
But still, like, again, I feel like Veruca Salt in the Chocolate Factory, where I just want more. There's,
Well, I mean, I think when we clicked on this paper originally, we really wanted to know like what cell types are being in vaccination, right? Where does the mRNA go, right, to
yeah,
produce a productive immune response? And because they were only doing their single cell RNA-seq after infection, it's too far of a time point to get that. At least that's my personal story with this paper. I was like, oh my God, I want to know how this vaccine works. And this tells us how it works in the context of like the infection, but I kind of just wanted to know before the infection, what's it doing? How is it priming the things to enter the infection? Yeah.
yeah. I was also kind of interested because, like, when you're being infected with it the first time, your entire immune system is learning and going through a learning process of understanding it.
Right.
And so I'm quite conscious that that's a factor with these mock-infected animals.
Mm-hmm.
And what would be interesting is if I could look at what the immune response is like immediately after vaccination. I
Mm-hmm.
mean, would we see things like granulocytes infiltrating? Would we see things like those interstitial macrophages, but maybe at a lower level?
Totally.
And then we could be able to pick apart what the difference is between, say, an immune response that's just to the vaccine or just to that spike protein and learning, and also the immune response that's corrupted by SARS-CoV-2.
Yeah.
And that'd be an interesting comparator to have, so we'd understand more underlying of like how the vaccines work, and then could give us insights into like how we want an ideal immune response to go.
Yeah,
And
no, totally. I totally agree with you. And I mean, all of that's hard work, and we appreciate the work that these guys have done.
yeah,
It's just that, like, there are so much. All good papers give you more questions, and seeing the techniques in action give you more ideas on other paths you could go down. And that's just the nature of science.
I mean, yeah, they sequenced every like RNA in a cell, not just one cell, but all the cells.
Yeah,
And I
yeah.
still, and this is something that would be unimaginable during my undergrad, but
Hmm.
now it's something
Hmm.
that is happening. And so
Totally.
instead of being happy with that, I'm a spoiled child. I'm like, no, no, I want more.
Okay, well, that's our coverage of this paper for this week. Join us next week for our news week, where we're going to survey papers to find something to cover in detail like we did this week, the following week.
Yeah,
All right.
so we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. So pipe up in the comments. Science is about thinking critically, and we're peer reviewing this paper. You can peer review us, I guess. And asking the right
Yeah,
questions
totally.
is an important part of it.
Hmm. Yeah, we peer review is a process that we can all participate in. So we hope that you have had a good time listening to us ramble on about microbiology today.